I am Mehrey. Today we are going to discuss a very important topic, which is CRISPR gene therapy, CRISPR gene editing. So by the end of this lecture, you will be able to know what is a CRISPR, what are the applications and benefits of a CRISPR. So let's start our topic by the basic question, what is CRISPR? First of all, I would like to talk about the full form of CRISPR. So what is the full form of CRISPR? CRISPR full form is clustered regularly shared palindromic repeats. So it is the full form of CRISPR. It is like molecular scissor for DNA comes from bacterium. It is a type of molecular system which scientists take from bacteria. Bacteria use the scissors as an immune system. When viruses attack bacteria, then they use this CRISPR to protect themselves from the viruses and damage their RNA parts, right? So scientists use this changed DNA in plants, animals, and humans. The application of this CRISPR is that scientists use it to change in plants DNA, animals, and humans, right? CRISPR, how CRISPR works. Now we are going to discuss that how CRISPR works. Number first, guide RNA. The CRISPR RNA guide find the exact DNA spot where we are going to discuss, where we are going to edit the gene. So guide RNA, it guide RNA and cut at the exact point of DNA spot. Number second, Cas or protein. The scissor that cut DNA. This is a type of scissor that which is helpful to cut the DNA parts. Right? After when we cut the DNA parts, then the DNA parts are naturally change their original position, and the scientists can add, remove, or change genes. Right? Example: fixing a gene that causes a disease. For example, scientists use this tool to modify a sickle cell anemia to know what are the reasons or which gene is caused due to which sickle cell is caused. So scientists use CRISPR editing to find out which gene within a plant is effective, right? So in the next uses of CRISPR, what are the uses of CRISPR? So number first, medicine. CRISPR, scientists use CRISPR as a medicinal tools. They use it as a medicinal tools, and in the pharmaceutical industry as well as to develop new drugs. Like scientists may know sickle cell anemia, and they try to know about that. What are the reasons behind it? Which gene is responsible for it? So they can see with the help of CRISPR gene editing. Number second, we have farming. As makes crop stronger and resistant pests. This is a very beneficial to make our plants protect our plants from pests. As we know, there are various types of pests which are going to impact or harm our crops. So while we using the application of CRISPR, we can able to protect our plants from different types of pests. Research, this is also a very important tool in research processes. We can use this tool, and we can modify different genes and their role in the life of anybody or any organisms. Future, we can predict our future. In future, we can make the genetically inherited disease medicines. We can easily make various types of medicines which may be positive to the various genetic disease, right? During using CRISPR, we have to know things to think about, right? Number first, we have scientists must be careful with human genes. When scientists are going to deal with the human genes, then they have to know the impact of this editing. Is there any negative impact of that? Scientists have to be very careful about it. about that. Ethics is very important. Not every change is safe. As we know that every change is not safe. When we talk about different Hollywood movies, then there are too many movies that are based on the information of biotechnology. Right? Last night I was looking a movie with the name of the movie was like in that story there a group of scientists or astronauts are going to discuss about is there any chances of survival in the Mars, so they can take a sample from the Mars and they are trying to observe them on their lab. After that, they observed that there is a small microorganisms which you observed, and after that they provide them energy, ATP source, etc. to know about the organism. That is there any that we can we live on the Mars? Is it possible for human beings to live on the Earth? So in this purpose they use various medicinal drugs like glucose, etc., and provide growth medium for the microorganisms, and they name them the microorganism as Cas9. So during their first investigation observation, they see that they are in that moving stage. After they provide them a growth medium and energy source. Then the Cas9 become big and big. After at the last of the movie, they are able to the microorganisms are able to kill all the astronauts, all the 6 groups of astronauts. So it is so while dealing with the gene therapy or biotechnology, we have to be very careful about all these things. Right. So this was all about today's lecture. I hope so. This is a clear cut. By the last, I would like to ask a simple question: that what gene would you like to edit? If you are able to edit a gene, then which type of gene you will select?